Myself Gazan. I am from Sacred Heart College. My topic is staining technique, differential staining technique. Gram staining. Gram staining technique is a differential staining technique used to differentiate bacterial species into two large groups. First one gram positive and second gram negative based on its cell wall composition peptidoglycan. The name comes from the Danish bacteriologist Hans Christian Gram who developed the technique in 1884, originally devised in 1882 but published in 1884. The crystal violet acts as a primary stain, iodine acts as a mordant, 95% ethyl alcohol or acetone act as a decolorizing agent, and safranin act as a counter stain. The mordant form the link between the cell wall of the bacteria and the primary stain crystal violet. Gram staining is still the cornerstone of bacterial identification and taxonomic division. Gram-positive bacteria rest decolorization due to thick cell wall peptidoglycan and appear violet in color due to the primary stain crystal violet. Gram-negative bacteria undergo decolorization due to thin cell wall peptidoglycan and take the counter stain safranin and appear pink in color. Decolorization step is an important step of gram staining process because the decolorization step distinguishes gram positive from gram negative cells. If the decolorizer 95% alcohol or acetone remains on the sample for too long, it may also decolorize gram positive cells. There are four types of staining. First one, primary stain, crystal violet, mordant, iodine, decolorizing agent, alcohol, counter stain, safranin. Gram-positive staining appears in violet color and negative in pink color. Based on shape, bacteria are classified into two types. They are cocci and bacilli. Gram-positive cocci, violet color and spherical shape. Example: Streptococcus species arranged in cluster and Streptococcus species arranged in chains. Gram-positive bacilli, violet color and rod shape. Example: Bacillus species, Clostridium species and Lactobacillus species. Gram-negative cocci, pink color and spherical shape. Example: Neisseria species. Gram-negative bacilli, pink color and rod shape. Example: Escherichia coli and Salmonella typhi. Gram-negative in pink color and gram-positive in violet color. Acid-fast staining is the differential staining technique which was first developed by Zeil and later on modified by Nelson. So this method is also called Zell-Nelson staining techniques. Nelson is in 1883 used Zell's carbon fusion and heat then decolorized with an acid alcohol and counter-stained with methylene blue. Thus, a Zell-Nelson staining technique was developed. Acid-fast staining method is used for those microorganisms which are not staining by simple or gram staining method, particularly the member of genus Mycobacterium, Nocardia, and Cryptospor- Cryptosporidium. The presence of thick waxy wall in the Mycobacterium species make penetration by stain extremely difficult. Once the stain penetrates it cannot be readily removed even with acid alcohol as a decolorizing agent. Because of this properly these organisms are called acid fast. Acid fast stain three different regions. The primary stain is carbon friction. The second stain is acid alcohol. In acid alcohol, 3% hydrochloric acid and 95% ethanol and is used as decolorizing agent. The final reagent is methylene blue stain act as a counter stain. First one, acid first. Bright red to intensive purple, red straight or slightly curved rods occurring singly or in small groups may appear beaded. Example, Mycobacterium, Nocardia and Cryptosporidium. Non-acid first. Blue color in addition background material should stain blue. Bacteria cultivated on blood agar may experience nutrient deprivation. resulting in a lower lipid content in the outer membrane, resulting in poor staining. So they are not recommended for acid fast staining technique. Acid fast staining. Acid fast bacteria red in color and non-acid fast bacteria in blue in color. Thank you.